Hello, dear students. Today we will be studying the topic cell, the unit of life. So we will be beginning with a few basics about how the study of cell started. The details about the cellular structure, we will be covering it in the next video. So let's start. So the cell study began with a man called Antony van Leeuwenhoek. He was a Dutch scientist. He developed a simple microscope using a biconvex lens and a pointed screw tip on one side. The object to be studied would be placed here, and he would place his eye on the other side. He was able to magnify objects up to 200 times by using his simple microscope. So, this enabled scientists of that era to study small objects in greater detail. This idea of a simple microscope led to the study of higher magnifications produced by using many multiple lenses. So next came Robert Hooke, an English scientist. He developed the idea of a compound microscope using two lenses, an objective lens and an eye lens. So the his idea was to use two lenses which would magnify objects to a larger degree, and he was able to magnify them roughly 2000 times. So the Robert Hooke's microscope gave the idea of the modern compound microscope which we use in the lab. Antony van Leeuwenhoek's microscope gave the idea of the simple microscope which we use for dissection purposes in the lab. But it's the common microscope which we use is based on Robert Hooke's microscope. Now, the compound microscope was able to use a beam of light for illuminating the object to be studied and produced a magnification of 2000 times. But after the compound microscope came the electron microscope, which used a fast beam of high energy electrons and magnified up to 2 lakh times. So a simple microscope, a compound microscope, magnifies spirogyra, a green algae like this. But if you see it under an electron microscope, you see it in far greater detail. The spiral chloroplast is very clearly visible. The compound eye of an insect becomes extremely clear. The body hair, the body structure becomes very clear under an electron microscope, but this is not so under a simple or a compound microscope. Now, as the study of cell structure, the microbiology advanced, then came the work of these three people: Matthias Jacob Schleiden, a botanist, Theodor Schwann, a zoologist, and Rudolf Virchow, a physician. These three people studied their own specific field and laid the foundation for the cell theory. Matthias Jacob Schleiden said that all plants are made up of cells, and cell is the structural and functional unit of all plants. Theodor Schwann studied the same thing, but said that all animals are made up of cells, and all the cell is the structural and functional unit of animals. He did this in 1938. He did it in 1939. And then came Rudolf Virchow. In 1958, he said all cells arise from pre-existing cells. So the three of their works were clubbed together in a cell theory that the cell is the smallest unit of structure of all living organisms. The cell is the smallest unit of function of all living organisms, and all cells arise from pre-existing cells. Now look at a human being. A human being has so many organs. Each organ system is comprised of these organs. The organs are made up of tissues. The tissues are made up of cells. It is the cell which will be functioning for that organ system. For example, for the nervous system, the neurons will be responsible for functioning of the nervous system. So cell theory suits for all organisms and all cells. Now the cells can be of various types. They can be single-celled, they can be few-celled, or they can be multicellular. The single-celled organisms are those which are having one single cell in their body. That single cell, whether it's in amoeba or whether it's in yeast or whether it's in bacteria, will be performing all functions of respiration, digestion, excretion, reproduction, every activity is done at the level of a cell. That's a single-celled organism. But when the activities are done by a group of cells, as in case of these green algae, Spirogyra, Wolffia, Volvox, they are all few-celled organisms. The activities are clubbed together by the group of cells. Now, in case of multicellular organisms like humans, donkeys, trees, plants, cats, any animal you take, any plant species you take, they all are multicellular. Now, in case of humans, the organism's body is divided into a number of organ systems. Each organ system is further made up of many organs. The organs are made up of tissues, and tissues are made up of cells. So there are millions of cells in the body of every organism. That's why they're called as multicellular organisms. Now, if you're studying the types of constitutions, you need to know the largest, the longest cells. The largest cells are those which are visible by the naked eye. They you can see, you can study them without using a microscope. Now, there are a number of large cells. One specific example I'll be mentioning here, it's a single-celled algae called Velonae ventricosa. This is called as the bubble algae. It can be as small as a pea. Sometimes it grows larger in size and can be held by the hand, can be viewed by the naked eye. Now, this algae is very large, but it is not the largest cell. The largest cell is the ostrich egg. This is the ostrich egg. And in the ostrich egg, it is the yellow part, which is the largest cell. Now, there was a bird called the elephant bird. This bird used to be the largest known bird species a couple of 100 years back. And this was the egg of the elephant bird. Due to overhunting, this bird went extinct. Now, at present, this ostrich egg happens to be the largest cell. This small little egg which you are seeing here, this is the egg of a hummingbird. So the hummingbird is the smallest of the bird. The egg is also the smallest egg in case of the bird kingdom. So the largest cell is of the ostrich, the yellow part of the yolk part. The white or the albumin part is only nutritive, protein-rich layer for the growth and development of the yolk. The yolk will form the fetus and will form the chick, which will hatch out later from the egg. Then comes the longest cells. The longest cells are the neurons. They can be as long as 3 feet. They can start from the base of the brain and reach till the lower extremity, or the base of the spinal cord and reach to the lower extremity. That means your feet. They can be longest cells. Very thin, but yes, the longest cells in the animals. Now, each cell in the body has its own specific shape, own specific size. For example, RBCs, they are biconcave, so that they can transport oxygen better. The muscle cells, they can be spindle shaped, so that they can function, contract, and relax better. The guard cells, they are bean shaped, so that they can open and close the stomata better. The bone cells are irregular. They can form that dense porous structure in a bone. The amoeba is irregular, so it can form food vacuoles and get its food faster and better. The WBCs are amoeboid, so they can engulf the incoming germs and bacteria and destroy them, performing immunity function better. So they all have developed according to their own specific functions. Then comes the eukaryotes and the prokaryotes. Now, what are eukaryotes and prokaryotes? For that, you need to know the word karyo. Here, it means nucleus. Eu means with nucleus. Pro means before nucleus. So eukaryotes are those which have a well-defined nucleus with a nuclear membrane. They have a nucleoli. They have cell organelles which are advanced and better developed, whereas a prokaryote primarily lacks a nuclear membrane. Cell organelles may be few and generally are primitive in nature, but the primary difference remains presence or absence of a nuclear membrane. So in a eukaryotic cell, there is a well-defined nucleus. Whether it's an animal cell or a plant cell, a well-defined nucleus with a nuclear membrane will be present in a eukaryotic cell. And in a prokaryotic cell, the primary difference remains that there is no nuclear membrane. So in a eukaryotic cell, you have a well-defined nucleus with a nuclear membrane. You have well-defined cell organelles. They are all absent in case of the prokaryotic cell. And there is no nuclear membrane. There are no nucleoli here. Now, you might wonder, we keep on talking about cell as microscopic, as very small. Why exactly are the cells so small? They have a small size so that they can achieve a greater surface area to volume ratio. Now, supposingly, to make our calculations easier, we assume that the cells are all perfect cubes. In any cube, the surface area is 6a square. The volume is a cube, where a is the side. That's the formula we use for calculation of the surface area and the volume. So supposingly we start with a cube of side 3 centimeter. The surface area will become 6a square or 6 3 square, that is 54 centimeter square. The volume will become 3 cube, that is a cube, or 27 centimeter cube. The surface area to volume ratio will become 54 by 27, which is 2. Now supposingly we cut down the two sides and the length and the breadth become 2 and 2 centimeter. The surface area now is 6 into 2 square, which is 24 centimeter square. The volume is 2 cube, that is 8 centimeter cube. The surface area to volume ratio becomes 24 upon 8, that is 3. Now, let's say we cut it down even further so that each side is only 1 centimeter long. Surface area becomes 6a square, which is 6 into 1 square, 6 centimeter square. Volume will be a cube, that is 1 into 1 into 1, that is 1 centimeter cube. So surface area to volume ratio will become 6 upon 1, which is 6. So largest surface area to volume ratio is achieved here. Now, what is the advantage of a large surface area to volume ratio? The large surface area to volume ratio, or that means the small size, will facilitate in rapid diffusion of gases, nutrients, and nitrogenous waste. Any metabolic waste can be removed from the interior of the cell and sent outward faster. It can repair a cell better. If the cell is injured, it can be repaired easily, and it will facilitate faster transmission of messages across a cell. If a cell has to relay a message from nucleus to the outside, from outside to inside, it will be relayed faster if the distance to be traveled is less. So that's all that we are going to study from this topic in this video. The remaining structural details of the cell we will do in the next video. Hope you have understood this. So wish you all the very best. Thank you very much, and God bless you all.